students, welcome back to our e-learning sessions of semester 5, subject pharmacognosy and phytochemistry 2. So keep your pen, paper and headphones ready. Let's start. Now, in this lecture, I will cover the lecture number 3. The topic is application of the latest technique, that is spectroscopy, in the isolation and purification of the crude drug. In the previous lecture, lecture 1, I have explained you in detail about the methods, that is modern methods of extraction. Then in my second lecture, part 1, I have explained you about the latest technique for the purification, that is chromatography. And again in my lecture 2, part 2, I have explained you about the rest of the techniques of chromatography in the isolation of and purification of crude drug. Now in this lecture 3, I am going to explain about the application of latest technique in spectroscopy in the isolation, purification and identification of crude drug. Now, what are the contents in this chapter? The main is the definition. I want to explain you about the spectroscopy in detail. What are the spectroscopy features? What are the applications of spectroscopy? What are the different types of spectroscopic methods that are used in purification and identification of the crude drug? What is the need of spectroscopy in pharmaceutical analysis? The main portion of spectroscopy is used in the pharmaceutical analysis. Then I will explain you about the importance of each and every method used in spectroscopy. Now, first, isolation and purification. As you all clear about the isolation and purification of crude drug, because in previous lectures I have already explained you about the isolation and purification. The main thing is that the isolation and purification of any compound depend on the physical and chemical characteristic of that compound which is to be isolated or separated. Okay. Now here in this lecture we will study various spectroscopic techniques like UV visible, IR spectroscopy, FTIR spectroscopy, mass spectroscopy, nuclear magnetic resonance spectroscopy. This is called as NMR. UV spectroscopic means ultraviolet spectroscopy. IR means infrared spectroscopy. NMR means nuclear magnetic resonance spectroscopy. So now, what is the need and what is the use of spectroscopy? Here we are studying about the phytopharmaceuticals. Phyto means plant and pharmaceutical means the constituent, the active constituent which obtained from plants is the phytochemical. These phytopharmaceuticals are pharmaceutical products composed of natural compounds from the botanicals instead of chemical. These phytopharmaceuticals are obtained from botanical means from herbal source. Now the most important problem for herbal phytopharmaceutical is that they are multiple component. Multiple means they have a mixed form of active constituent. So it is not easy to do analysis of the compound. So for proper analysis we have to understand the chemistry of phytoconstituent, which constituent is present in that. For this, the spectroscopy is the useful method based on the characteristic, isolation, structural identification and quantification. We have to plan according to their characteristic. We have to plan the spectroscopic analysis. First, we have to look at the chromatographic techniques which isolate the compound. With the help of chromatographic technique, we will isolate first we will extract, then we will, with the help of chromatographic technique, we will isolate the compound up. Once the components are separated or isolated, then they can be analyzed by the help of spectroscopic methods. Got my point? That first we'll do extraction, then we will use chromatographic techniques for the isolation, and for the analysis we are using spectroscopic method. This is the introduction. Now, what is spectroscopic analysis? Analysis is the series of processes in order to identify a substance, components of a solution or the mixture or the determination of structure or the structure of the chemical compound. Two types of analysis can be done. One is qualitative analysis, other is the quantitative. Quantity means how much, and quality means each provides the information about the identity, its atomic or molecular species or functional group, about the quality of the compound. And in quantity, it gives about the one or more component present in how much amount. This all depends on the quantitative analysis. Now next, what's the need of phytopharmaceutical analysis? The main important parameter for the need of phytopharmaceutical are the three main points: identity, purity, and content. Identity means the condition of being the specific drug, the correct herb. Purity means it should be free from contaminant, it should be free from adulterant. And content means the amount of active constituent present, because this amount will decide the yield of the product. Okay. Next, what are the steps? How do we do the phytopharmaceutical analysis? So the major steps are: first we prepare the sample. After preparing the sample, we will do chromatographic analysis like isolation and purification. Then we will do identification of the analyte by spectroscopic method, and then we will do quantification, how much yield we will get. Now, the major portion is modern methods used in the identification of crude drug, or we can say different spectroscopic method. Here we are discussing about four important methods. One is UV spectroscopy, ultraviolet and visible spectroscopy. Next is infrared spectroscopy, IR spectroscopy. Next is mass spectroscopy. And fourth is NMR, nuclear magnetic resonance spectroscopy. Now, spectroscopy is the branch of analytical chemistry. As I have already told you, it is used in pharmaceutical analysis. So it is the branch of analysis. And what spectroscopy is doing? It is dealing with the study of interaction of electromagnetic radiation with the matter. The most important of such interaction is that energy absorbed or emitted. The principle behind spectroscopy is energy is absorbed or emitted by the matter in discrete amount, in different amount, that is called as quanta. Solvent used basically in spectroscopy is the 95% alcohol. And the example, for example, in spectroscopy we are using UV. So electronic transitions of atom and molecules are observed by the principal Lambert-Beer law. In IR region, molecular vibrations is observed, that how molecular changes from one energy state to another. Next, first we'll learn the UV spectroscopy. UV is ultraviolet spectroscopy. So you should note down one point, that UV spectroscopy obeys the Lambert-Beer law, which states that when a beam of monochromatic light is passed through a solution of an absorbing substance, the rate in decrease the intensity of radiation with thickness of the absorbing solution is proportional to the incident radiation as well as the concentration of solution. For UV spectroscopy, we have to prepare the dilutions of different concentration. So a ray passes through this solution, and this is the formula for Lambert-Beer law application, where we have to find out the radiation, the absorbing substance. Okay. Now, the absorption spectra of plant component can be measured in, I have told you, various dilute solution against a blank solvent. And UV spectrophotometer is an automatic recorder. For UV spectroscopy, the range for colorless compound is 200 to 400. For colored compound is 200 to 700. So the wavelength of the maxima and minima in this spectroscopy is measured in nm, and the intensity of absorbance, that is optical density, at the particular maxima and minima should be recorded. Now, what are the applications? Why we are using UV spectroscopy? It is very useful in measurement of different plant constituent. It is also useful for the presence of polyacetylines, aromatic compounds, for monitoring the evaluated of various chromatographic techniques for the purification of plant products. The complete absence of UV absorption. If some compound does not have the ability to absorb the UV rays, then also we get some information regarding the structure. And it also helps in indicate the presence of saturated lipids, alkaline lipid fractions, organic acid, aliphatic amino acid, sugars, and quantitative determination of glycyrrhizinic acid in crude drug can also be measured by the help of UV spectroscopy. We can also do the different curcumin estimations. Curcumin we have obtained from turmeric. By the help of UV spectroscopy, we can also do the estimation of curcumin. Now, next comes IR spectroscopy. IR means infrared spectroscopy, and infrared spectroscopy is also depends on the electromagnetic radiation, and the main three ways by which IR spectroscopy can generally be analyzed are: first, measuring reflection, emission and absorption. This spectroscopy is generally, I have already explained you before that, on the absorption and the emission. These two are the main criteria, and the major use of IR spectroscopy is to determine the functional group of the compound. Whenever we want to determine the functional group of compound, we can use IR spectroscopy. These are the diagram for IR spectroscopy. This is the visible range, UV and IR. This is the detecting rays. Now, how can we explain the IR? IR, the reason which extend 80 angstrom to 35,000, 8,000 angstrom to 35,000 angstrom wavelength is known as IR region. This gap of wavelength is known as IR region. IR radiation are associated much lower energy as compared to UV, and IR may be measured on plant constituents. IR can be measured for plant constituents in IR spectrophotometer, either in the chloroform, CCl4, or solid state like PKBr, means potassium bromide. IR is also used to gather the information about the structure, and is an important tool for the purity of check the purity of the compound. So we can conclude that IR can be used for the structural elucidation, for determining the functional group which are encountered in different plants. Now, the reason in IR spectroscopy above 1200 centimeter shows spectral bands or peak due to vibration, and below 200 bands due to vibration of the whole molecule, and because of this is called as fingerprint region. And the bands which we obtain in IR denotes by 3: S, M and W. S is strong, M is medium, and W is weak. So these are the two ranges, IR spectrum above 1200 and IR spectrum below 1200. Now, with the modified form of IR spectroscopy is FTIR, Fourier transformed IR spectroscopy. It is termed as time domain spectroscopy because the changes in the radiant power are recorded with changes in time. This is more advanced form of IR spectroscopy. It is the simple or special instrument which make use of multiple detectors. In this, multiple detectors are attached, so we'll get the result in very short time. For example, FTIR spectroscopy can be used for the chemical stability of herbal extracts. Next, our third one is mass spectroscopy. It is an instrumental technique in which the sample is converted into rapidly moving positive ions and then separated and characterized. Here, the role of ions. Mass spectroscopy generally produce charged ion, consist of parent ion and ionic fragments of original molecule, and sorts these ions according to their mass charge ratio. So mass spectroscopy differentiate the compound according to their mass and charge ratio. Got my point? Next, what are the uses of mass spectroscopy? First, to determine the molecular structure, molecular weight. This technique is also utilized for the low molecular weight plant constituent, also used in peptide analysis. And the sample or amount for detection used in mass spectroscopy is in micrograms. And in microgram, it can provide an accurate molecular weight of any compound in the complex mixture also. For example, analysis of micro capsules containing cinnamon leaf oil and garlic can be estimated with the help of mass spectroscopy. And this is the figure for mass spectroscopy. This is the sample inlet. Source means gas phase ions. Here ion sorting is done by the analyzer. Ion detection is done by the ion detector. And this is the data system attached to the full data and detector system, where we get the chromatograms and different data for the extracts or crude drugs. So here you get the clear idea about the mass. This is inlet, means we have to inject the sample. Source means gas phase ions. Then ion sorting is done. Then ion detection is done. Now the last is the NMR spectroscopy. This spectroscopy involves the magnetic energy of nuclei when they are placed in a magnetic field, and transition occur in the wave region of respected. So the study of radio frequency radiation, means radiation by the frequency of nuclei, is called nuclear magnetic resonance. The name only elaborated the definition. Now, what is NMR spectrum? It generally gives an idea about the structural background of any compound. 13C NMR is generally used to identify the number of carbon atom, and 1H NMR is generally used for how many type of hydrogen atoms are present in our compound. So different types of NMR can be used: 13C NMR, 1H NMR. And what is the role of NMR? How the procedure can be done? In this, the sample of a substance is placed in the solution in an inert solvent between the poles of powerful magnet, and the proton undergo different chemical shift according to the main important part, according to their molecular weight or molecular environment. This is measured in NMR apparatus with the help of inert compound like TMS, trimethylsilane. Okay. Now, NMR spectroscopy is generally used in the phytochemical analysis, structural elucidation, active constituent of, for example, Terminalia cellula powder. We can also show the presence of tannins, alkaloids, protein, carbohydrates, various phytosterols with the help of NMR spectroscopy. And nowadays, proton NMR spectroscopy are used by various phytochemists as a fingerprinting technique, as a marker detection for the detection of the marker compound. And the isolated compound was confirmed by, so we can conclude that the isolated compound was confirmed by UV, IR, mass and NMR spectroscopy. What are the applications where NMR spectroscopy is used? Proton NMR spectroscopy is used for the determination of the structure, and sometime in many compound hydrogen atoms are attached in different groups and different situations. In this case also the NMR spectroscopy is used, and the combination of two techniques, 13C NMR and the NMR, this gives a powerful means of structural elucidation for the new compounds or the multiple components like alkaloids, flavonoids, terpenoids. And it is useful for the analysis of glycoside in indicating the linkage between aglycone moieties, between sugar moieties and their configuration. And can also different types of extracts and powder can also be isolated and identified by the help of NMR spectroscopy. So at last we can conclude that the spectroscopic techniques show rapid and precise, means it shows the result in accurate form, precise form and in less time consuming. So the time consumed in this type of techniques is very less. So this is a very good technique for the analysis of phytopharmaceutical as compared to chemical analysis and trigonometric analysis in our old method. What are the important questions you have to cover for these topics are discuss in detail about the different applications of spectros. Exam question may come like this: what are the different application which can be used for the analysis of phytoconstitution? So if any question come regarding the spectroscopic analysis, you can write these 4 methods. Then what is the role of IR spectroscopy separately can also be asked. Mass spectroscopy separately can also be asked. So you can be ready, you all should be ready for these questions. These are the bibliography and the references from where I have taken the whole matter. And NPS Sanger, the textbook of pharmacy, is also very good for this topic. Now, thank you. Be safe. Stay healthy. Bye. All the best.